Our next speaker is Dr. Teresa DiLorenzo. Teresa has not only pioneered a robust basic research program, but she's also built translational bridges between scientific disciplines here at Einstein and elsewhere. From her roots in the Stan Nathanson Laboratory, and Stan is another Einstein member of the National Academy, to clinicians in the Diabetes Center, Teresa has added her own brand of enthusiasm and focus on finding definitive treatments for type 1 diabetes that Vern just alluded to, so you better work a little faster. But while the last 2 decades have seen enormous advances in insulin treatment and equally remarkable achievements in understanding the immune basis of type 1 diabetes, they have not yielded, as of yet, definitive therapies with the potential to prevent or cure this disease. Dr. DiLorenzo's work has built on the tenet that the subset of CD8 positive T cells that recognize autoantigenic peptides bound to class 1 MHC molecules expressed on the insulin-producing beta cell are absolutely required for the development of type 1 diabetes. And parenthetically, I should point out that in the late 70s, it was here at Einstein that Norm Fleischer and Sunil Shin first showed that T cells are indeed the culprit in the beta cell destruction in type 1 diabetes in the mouse. Dr. DiLorenzo's studies to identify the beta cell antigens responsible for the autoimmune response, therefore, could lead to truly specific therapies for type 1 diabetes. Teresa is associate professor in both departments of microbiology and immunology and of medicine, and the Irma T. Herschel Career Scientist Awardee. Teresa?
I want to thank the symposium organizers for giving me this great opportunity to highlight the work, some of the work that's going on in our lab today. So type 1 diabetes is an autoimmune disease in which the insulin-producing beta cells are the target and they are ultimately destroyed. And the disease carries with it a variety of devastating complications. And it's characterized by infiltration of the islets by leukocytes or inflammatory cells, and this process is known as insulitis. And the beta cells are ultimately destroyed due to autoreactive T cells. As an autoimmune disease, it results from a failure to establish and/or maintain self-tolerance to particular antigens. And there are multiple genetic loci and multiple ill-defined environmental factors that contribute to susceptibility to type 1 diabetes. This is what the disease looks like histologically. So this is a normal islet in which, in this particular preparation, the beta cells are stained purple. So this is a normal islet, but what happens in a diabetes-prone individual is that islets are slowly attacked by cells of the immune system. Here you can see those cells at the periphery of the islet, but they eventually penetrate into the islet, and you can see that they're destroying the purple beta cells as they go, until you get to a point where you have islets where all of the beta cells have been eliminated. And at the point where approximately 80% of the beta cell mass has been destroyed, that's when the patient or the animal will show clinical signs of type 1 diabetes. Now, if we look more closely at this infiltrate, we would actually see a very complicated picture. In fact, nearly every cell of the immune system participates in some way in the autoimmune process that leads to type 1 diabetes. And I clearly don't have time to go through every cell and what every cell does, but just to make the point that some cells participate to lead to the demise of the beta cells in the pancreatic islet, and some cells actually serve to slow the destruction of the beta cell. So for example, T cells known as regulatory T cells serve to preserve the beta cell. They dampen the impact of effector T cells that can destroy the beta cell. And 2 types of T cells that participate in the demise of the beta cell are the CD4 T cells releasing Th1 type cytokines and CD8 cells. Our lab focuses almost exclusively on CD8 T cells, and hopefully you'll see why. And so I'll be concentrating on these cells today. These cells can directly recognize antigens presented on the surface of the beta cell, and they can kill the beta cell via a variety of mechanisms. Now, the way the CD8 T cell recognizes its antigen is shown here. So this is a CD8 T cell that has on its surface an alpha-beta T cell receptor and also a CD8 co-receptor. The antigens that are recognized by CD8 T cells take the form of peptides that are presented by class 1 major histocompatibility complex, or MHC molecules. And these molecules are composed of a light chain known as beta-2 microglobulin and a heavy chain with multiple domains. And of most interest for us is that the alpha-1 and alpha-2 domain you can see form a peptide binding groove. So class 1 MHC molecules present short peptides from 8 to 11 residues long that are derived from beta cell proteins. And in type 1 diabetes, some of these peptides are targets that are recognized by T cells and lead to the ultimate demise of the beta cell. Much of what we know about the importance of CD8 T cells in type 1 diabetes has come from this mouse model of the disease. NOD mice spontaneously develop autoimmune diabetes. Diabetes is first seen in these mice at approximately 10 weeks of age, but the insulitic process starts very early and can be detected at 4 to 5 weeks. Most in the field believe that the NOD mouse is a very good model for type 1 diabetes because it shares many characteristics with the disease in humans. Beta cells are destroyed due to an immune attack. The disease in both humans and in the mice is polygenic. Again, there are ill-defined environmental factors that can influence diabetes incidence in NOD colonies that are located at different institutions. And of particular importance for the talk today, you'll see that some of the antigens recognized by T cells in the NOD mice are also recognized by T cells in patients. Now, we focus on CD8 T cells, so I need to justify to you why we do that. Studies in the NOD mouse have shown that these T cells are absolutely required for disease development. If you ablate these T cells, either by treatment with a depleting antibody or if you genetically block their development by knocking out either beta-2 microglobulin or the CD8 co-receptor, diabetes does not develop in the mice. Transfer studies have also been useful in indicating to us that CD8 T cells are required for the development of the disease. So if one isolates T cells from pre-diabetic NOD donors, you find that you need to transfer both CD8 and CD4 T cells to a recipient in order to adoptively transfer disease. And finally, we and others have isolated CD8 T cell clones from the islets of NOD mice, and we've shown that these T cell clones are cytotoxic to beta cells in vitro and that they can accelerate disease in the setting of transfer models or T cell receptor transgenic mice. But what about in humans? I mean, today is all about going from mice to men and back again. So it turns out that in humans as well, CD8 T cells are suspected contributors to beta cell damage. For example, CD8 T cells are prevalent in the islets of new onset and graft recurrent type 1 diabetes patients. And we and others have shown that there are CD8 T cells specific for beta cell antigens in the peripheral blood of type 1 diabetes patients. So given the importance of CD8 T cells, we feel that they constitute an attractive therapeutic target. So in order to devise an antigen-specific immunomodulatory therapy for this disease, we first need to identify the peptides that are targeted by the CD8 T cells in the disease. And you will see, I'll give you an example of how we do this, and then you'll see how this process can lead to the development of reagents to manipulate the T cells, and we hope to get as far as Vern has done and ultimately get to a therapy, specifically an antigen-specific immunomodulatory therapy. So ideally, we would want to identify the targets of the beta cell specific T cells in patients themselves. However, this poses a number of problems which are shown on this slide. First, there is essentially no access to the islet infiltrating T cells in patients. It is also difficult to identify patients with early encephalitis, and those are the T cells you would really want to study because those are the T cells that are initiating the disease, but you don't have good access to those in patients. And finally, even if you study T cells in peripheral blood, there are only limited numbers of CD8 T cells available, particularly from the pediatric type 1 diabetes population, which is the population that we study in collaboration with the Children's Hospital at Montefiore. So to overcome this problem, there are several possible solutions, but the one that we've been banking on is to use NOD mice that are transgenically expressing human class 1 MHC molecules, or HLA molecules. So these are humanized mice. And we're using the islet infiltrating T cells from these mice to allow us to identify peptides that are recognized in the context of human class 1 MHC molecules. And then once those peptides are identified, we can query patient samples and determine whether those peptides are relevant to the human situation. So I just briefly want to show you how we get these islet infiltrating T cells. So this would be an NOD mouse transgenically expressing a class 1 MHC molecule. We isolate the islets from these mice by dissociating the pancreas with collagenase, and these are 3 isolated pancreatic islets. And my student Irene actually made a movie that shows when you put these islets into culture in the presence of the T cell growth factor IL-2, the inflammatory infiltrate, mostly T cells, exits the islet and can then be studied. So this is the inflammatory infiltrate just storming out of the islet. This movie is taken over a course of about 3 hours. So now that these cells are free from the islet, we can study these cells and use them for antigen recognition assays. So the 2 main antigens that are under study in the lab at the moment are this antigen known as IGRP. This is, we refer to it as our antigen. It's near and dear to our heart because our lab discovered this antigen in 2003. We discovered it as being prevalently targeted by T cells in standard NOD mice. And another antigen that we study is insulin, which is known to be a very important T cell antigen in both NOD mice and in patients with type 1 diabetes. So what we do now is we take the T cells from the humanized mice, we use those T cells to scan peptide libraries of IGRP and of insulin, and in that way we can identify the specific peptides that are recognized by the T cells. So this is a summary of actually quite a bit of work. In this case, we used NOD mice that were expressing the human class 1 MHC molecule HLA-A0201, and we used a T cell recognition assay known as a gamma interferon ELISpot. The details of that are not important. All you need to know is that on the y-axis, this represents T cell activity, and each symbol represents T cells from an individual mouse. So what you can see is that we were able to identify 3 peptides from IGRP and 3 from insulin that are recognized by T cells in the mice. But now the key question is, are these peptides relevant to the human disease? First of all, are the sequences conserved? So I wouldn't be telling the story if there weren't a reasonably good conservation of the peptide. So you can see that this middle IGRP peptide is 100% conserved between mouse and human, and the other 2 peptides differ by only 2 residues. And I'm very happy to report that now we and others have shown that both IGRP 228 and IGRP 265 are recognized by CD8 T cells in patients. So it really blesses the strategy, and it does support the idea that we've been trying to sell for several years now, that humanized mice are a good way to identify disease-relevant peptides. And there haven't been human studies done using the peptides we identified from insulin, but you can see two of them differ by only a single residue if you compare the mouse and the human peptide. So that just gives you an example of how one goes about identifying the peptides targeted by these T cells. And now I want to describe to you work that we're doing on an antigen-specific immunomodulatory therapy for the disease. So just a little more immunology. So dendritic cells of the immune system are very important in initiating an adaptive immune response to a pathogen. So when these dendritic cells are exposed to microbe-associated molecules, they undergo a maturation process in which they upregulate MHC molecules and they upregulate T cell co-stimulatory molecules. So when a T cell encounters such a dendritic cell, it will be activated and it will be ready to fight the particular offending pathogen. However, in the steady state, that is, in the absence of infection, dendritic cells are constantly processing and presenting self-antigens to T cells in a way that leads to T cell tolerance to self-antigens. And this tolerance can take the form of T cell deletion or actual elimination of self-reactive T cells, T cell anergy or non-responsiveness, or the development of regulatory T cells, which I mentioned earlier. So we reasoned that if we could target beta cell antigens to steady-state dendritic cells, we might be able to promote tolerance to those beta cell antigens. So how to target antigens to dendritic cells? Nussenzweig and Steinman at Rockefeller had shown that foreign antigens could be targeted to dendritic cells using an antibody to a dendritic cell surface receptor known as DEC205, and this molecule is depicted here. So they found that by coupling an antigen to an antibody to DEC205 and then treating an animal, T cells specific for that antigen would be tolerized. So they would proliferate initially, but that initial proliferation would be followed either by deletion or non-responsiveness. So we attempted to ask whether a beta cell self-antigen could similarly be targeted to dendritic cells, and we asked what the impact on T cells would be. The way that the anti-DEC205 is thought to deliver antigen is that it binds to DEC205, and DEC205 likes to spend its time in intracellular compartments. And when it does so, it carries with it the antigen bound to the antibody. And via a process that's poorly understood, that antigen ultimately escapes into the cytosol, is degraded by this cytosolic protease to peptides, and the peptides from the antigen, shown in red, ultimately find their way onto class I MHC molecules and are sent to the surface of the cell. So here's what we did to determine whether beta cell antigens could be targeted to T cells using this approach. We first began with T cells isolated from a transgenic mouse expressing the T cell receptor of a beta cell specific clone known as AI4. We took these CD8 T cells, labeled them with CFSE, and then transferred them to an NOD mouse. We waited 1 day, and after 1 day, the mice were injected with anti-DEC205 that was coupled to a mimetope peptide known as MIM-A2, which is recognized by the AI4 T cell clone. We then isolated spleen and lymph node either 3 days later or 12 days later, and by flow cytometry we examined the fate of the transferred cells. So they could be easily identified because they were double positive for this congenic marker and for CD8, and they were also labeled with CFSE. So if we saw CFSE dilution, we would know that the transferred T cells had proliferated. So the results are shown here. We looked at the transferred cells in 3 different sites in mice treated with the anti-DEC205 antibody, and you can see that there is CFSE dilution or CFSE low population in all 3 sites examined. So this said that the T cells were seeing their antigen and were proliferating in response to that antigen. In contrast, in mice treated with an isotype control antibody or with PBS alone, you see only essentially no proliferation in spleen and peripheral lymph nodes. You do see proliferation in pancreatic lymph nodes in all cases because that's where the natural beta cell antigen recognized by AI4 is presented. So everything looked good. We knew that we were getting T cell proliferation, but now we asked, what is the fate of these T cells? What is the ultimate fate of the T cells? And much to our delight, we found that after 12 days, these beta cell specific T cells are actually deleted. So if you look for these T cells, which should be present in this purple gate, they're essentially not present in the treated animals, whereas they are present in the isotype treated control animals. But you may be saying, well, maybe the cells are not really deleted. Maybe the cells are actually all in the islet. Well, we examined the islets from these same mice, and we found, fortunately, that the T cells are not in the islet either. They have truly been deleted. You can see that they are concentrated in the islet of a control-treated animal. But there is a lot more work that needs to be done because these are transferred cells. What about endogenous beta cell specific T cells that arise in an NOD mouse? I have some exciting, although very preliminary data to show you on my last data slide. So in this case, we wanted to quantitate T cells specific for IGRP in NOD mice because I told you IGRP is targeted by a prevalent population of T cells in NOD mice. So NOD mice were treated with either isotype or anti-DEC antibody linked to another mimetope peptide NRPV7, which is recognized by IGRP-specific T cells. So we treated mice at 4 weeks of age, isolated their islets at 8 weeks of age, and then IGRP-specific T cells were quantitated using peptide MHC tetramers. And again, this is very preliminary, but I think very exciting data. Each symbol here represents an individual mouse. So we've examined 2 mice that have been treated with the isotype control antibody, and you can see a high proportion of their CD8 T cells are specific for IGRP. In contrast, in the animals treated with the anti-DEC205 antibody, there's a considerable reduction in the proportion of these T cells. This is exciting to us because this is occurring in the context of ongoing autoimmunity in NOD mice and in mice that are known to have CD8 T cell tolerance defects. So that suggests that this method might be applicable even to diabetes-prone individuals who might have similar defects. So the 2 main conclusions are that T cells from HLA transgenic NOD mice recognize epitopes of beta cell antigens that are clearly relevant to the disease in humans, and I showed you the example of IGRP. And we think that in vivo targeting of dendritic cells with beta cell antigens is an exciting approach that could lead to reduction of both transferred and endogenous autoreactive CD8 T cells. I just want to thank my whole lab for their efforts and support. In particular, I want to highlight Irene, who did some of the work with the HLA-2 transgenic mice, and she also did the patient study where she tested patient samples for reactivity to IGRP peptides. And I also want to highlight postdoc Oronika. She did all of the DEC205 work that I showed you today. Very promising project. She's done an excellent job. And I also want to thank my outside collaborators, Dave and Michelle, for the development of the HLA-2 transgenic mice, Ralph and Kristen at Rockefeller for reagents and advice concerning the DEC205 project, Per Santamaria at Calgary for peptide MHC tetramers, and my funding agencies. And finally, all of you for coming out this morning and for your attention. Thank you.